I say this has relatively limited use in wildlife populations, and that's at least for one reason. We can all imagine that there are other sources of variation that could cause parents to look like their offspring. Maternal effects being the obvious example in mammals. Also, it doesn't make very efficient use of all your genetic relationships because you're only comparing one sort of genetic relationship. You're comparing parents to offspring. Uncles, sisters, grandparents all share genes with other individuals too. So ideally, we want to use that information to give us better estimates. So typically what we would use now is what's called the animal model. And I put this quote up here by my former boss, Luska Kruk, who is one of the smartest people that I've ever met. And Luska acknowledges that quantitative genetics has an ugly name. And I put this up because I think a lot of people are put off by it because a lot of the terms are kind of opaque the first time. Quantitative genetics in general and the animal model. But because as evolutionary change within a population requires a genetic basis, it's a subject that is integral to many ecological questions, arguably all evolutionary ecology questions. What the animal model essentially is doing is using a mixed effects model, which are in relatively common use in ecological research now when we have structuring our data. For example, if we have multiple records on an individual, we would put individual as a random effect in our mixed effects model. All the animal model is doing is breaking up that individual effect into the underlying components, such as genetic variants, maternal effects, etc. Essentially, what it's doing is using the pedigree for the population to inform how much, or infer how much of this variation in phenotypes is due to genetic relationships. Do animals that are genetically similar also resemble each other phenotypically? Give you a bit of an appreciation for estimates of variability in wild populations. These is far from a comprehensive survey, but just a few examples that I gleaned from the literature in wild mammals. You can see the traits that you would sort of expect would be highly influenced by environmental variation, such as body mass and soy sheep, relatively low heritability. So 13% of the phenotypic variance is comprised of a genetic component. More structural traits, however, tend to have a higher heritability. So about 70% of the variation in horn length of a bighorn sheep is due to the underlying genetic contributions. First step in carrying out an animal model-based analysis is to build your pedigree. The way we did that with the ground squirrels is to use our maternal links, just through observations. We can observe the offspring and the moms in the field, or they'll give birth in the lab. The females will mate multiply, however, so we had to use molecular paternity identification to determine the paternal links. If you've seen a pedigree in a textbook, it tends to look something like this. Females and males linked to their parents. This is what the pedigree for our ground squirrel population looks like. It's a mess, I know, it looks a bit like a bird's nest, but each individual here is a white entry. Females are linked to their offspring with a yellow bar. Males are linked to their offspring with an orange bar. What I'm showing is that there are 7 generations and lots of cross linkages across all the individuals, creating lots of diversity of genetic relatedness values, in essence, lots of power in our analysis to be able to pull out those genetic estimates in our phenotypic variance measurements. The actual animal model that I fit was essentially just an extension of the conceptualization of genetics and environment influencing phenotypic variation. I focused on additive genetic variation because that's the variation that's passed linearly between generations, dominance isn't. And I split the environmental variance just into more ecologically relevant sources of variation. So I fit a maternal effect that I was able to estimate by comparing mothers to their offspring and also dads to their offspring because there obviously aren't any maternal effects along the paternal lines. And the permanent environment effect just refers to non-genetic sources of repeatability. So if an animal is on a high-quality territory, it may breed or emerge earlier every year. That isn't due to underlying genetics. It's an environmental source of repeatability. And error residual variation just will encompass the other unexplained variation in the model. In the model, I was able to detect reasonable levels of additive genetic variation, relatively low maternal effects, and relatively low non-genetic repeatability. Put that together into our heritability analysis. comes out to 0.22. So 22% of the variation that we see in terms of when an animal emerges from hibernation is due to underlying genetic variation. Early parents tend to give rise to early offspring. It does have evolutionary potential. The second step was then to look at how much of the association between emergent state and ester state is due to an underlying genetic linkage. The way that I did this was to fit a bivariate animal model to estimate both the heritability of emergent state again, the heritability of ester state. In both of those cases, they're relatively similar, in both cases significantly heritable, and how much of this phenotypic variation was comprised of an underlying genetic correlation. And it was relatively high. So close to 1, in fact, that we would suggest that the same genes that influence emergent state are influencing reproductive phenology. So under that objective, we were able to establish that there is individual variation when they emerge. It has a heritable genetic component and a genetic association to when they start mating. The second step was then to look at what are the environmental influences on emergent state. And sort of underlying or behind this objective was an interest in whether or not hibernation phenology could be influenced in the same way that a number of other phenological traits are being influenced by climate change. So we know that lots of plants are flowering earlier now. Same thing with many migratory birds. They're arriving from migration and breeding earlier. But with few exceptions, we really have no idea if this is the case for hibernating animals. And of concern, populations of birds that haven't shown a phenological response to climate change are more likely to be in decline. And I think we could predict both the responses to climate change, the advances, as well as the declines, if we think back to the phenological trait as a component of the seasonal cycle of the animals. So ideally, again, what the animals are trying to do is synchronize their reproduction with the seasonal availability of resources. For example, if the leaves of an oak are flushing earlier, the insects are declosing earlier, the energetic period, the favorable energetic period is advancing. What the insectivorous birds are then trying to do is keep in march or lockstep with their environment. If they aren't able to do that, they're then desynchronized with their environment. Individual fitness is likely to decline, populations are likely to decline. So we asked, how are the emergent states of Columbian ground squirrels influenced by weather variables? And have any weather variables exhibited any temporal trends over the past 20 years? The two weather variables that we looked at were ambient temperature and snow cover. This again speaks to how hibernation phenology could be different than some of these other phenological traits. For example, snow cover may not be a direct influence on the phenology of birds, but these guys are hibernating a meter under the ground, or pardon me, a meter under the snowpack. A snowpack that is delaying the flush of their forage plants, making it hard to thermoregulate, etc., etc. And also, snowfall in this part of the Rockies is highly dynamic. This was a picture I took of our field site, 2009. This is the field crew going home at the end of the day. This is what it looked like 2 days later. Big dump of snow. We were starting to see the odd squirrel trickle up. This totally knocked them back by about a week or so until the snow started melting. The way that I did this was to estimate the phenotypic plasticity of ground squirrels in response to environmental variation. Phenotypic plasticity is defined as the differential phenotypic expression of a genotype or an individual across an environmental gradient. That could be ambient temperature, for example, and emergent states. And I estimated the reaction norm, of the individual emergent state to changes in these weather variables. The reaction norm is simply the slope of the individual relationship between the environmental variable and the phenotype. Steeper slopes, more plastic individuals. I was able to get the temperature data from the National Climate Archive that Canada records from a weather station about 50 kilometers from our field site. Used a sliding window approach to determine which period over which to estimate ambient temperature or average temperature. Not surprisingly, the window told me that it's the 12th of April to the 3rd of May. right around the time the animals are gearing up to or are emerging. For snow-free day, or for snow cover, what I did was take the first day of the year that had 0 snow on the ground. However, if we did get one of these snowfalls before all the animals had emerged, I simply took the first snow-free day after that. So we can see here in 1992, relatively early snow melt. March 31st was the first day with 0 centimeters of snow on the ground. In 2009, we can see this snowfall showing up in the data set here. Snowfall date was almost a month later than in that previous year. So ground squirrels did exhibit phenotypic plasticity in response to both weather variables. Here we have mean-centered environmental variables on the x and y-axis, and then emergent state on the z-axis. These are mean-centered, and each of these little circles represents the mean for all the females in a given year. Each one of these circles represents a year. So the females did emerge later, about a day and a half later, with each 1 degree decrease in temperature, and about a tenth of a day later with each 1 day delay in snowmelt. So the second thing was to ask whether or not either of these variables have exhibited a temporal trend over the past 20 years. And the answer with respect to spring temperature is no. At least within this one window, at this site, there hasn't been a consistent trend in ambient temperature. The result with respect to snow cover is quite different. And it's actually become later. And that's because those late season snowstorms, like I just showed you a picture of, have become more and more likely over the past 20 years. Whereas in the first decade of the study, we only had 1 significant snowfall event after the middle of April in 1 year. In the most recent decade, we've had 7 such events. And in essence, what that has done is caused the date of snowmelt to become later by about 3 days a year over the past 20 due to these late season snowstorms. And so if we put these two things together, a documented phenotypic plasticity to snowmelt dates, a temporal trend in snowmelt date, we get a phenotypic trend in the ground squirrels that runs counter to almost all the phenological traits that are reported in the literature. Whereas most things are getting earlier and earlier and earlier, presumably due in large part to warming temperatures, the ground squirrels have actually become consistently later over the past 20 years, by about a half a day a year. So that means if we say half a day a year over 20 years, they're essentially emerging now about 10 days later than when the study started. Remembering again that that's 10 days of a 3 to 4 month chunk, so it represents a significant proportion. So the second question was to ask that if these animals are being pushed later and later, what are the fitness consequences of this? We know for certain that a ground squirrel can likely emerge too early. If a ground squirrel was to emerge around Christmas, it wouldn't be a very happy one. They would probably find themselves in the red part of that curve, no food to eat, very cold, very wet, hard to thermoregulate, hard to stay alive at that time of the year. What we wanted to ask is, can they also emerge too later? Specifically, if reproduction is timed so as to be at an optimum time of year, and they're being pushed later than that, how is that influencing the fitness of the individuals? To calculate fitness, I made a composite measure of the overwinter survival of females, 1 if she survived basically a year after we reported that emergence, and half of her number of surviving pups. We discounted the number of pups by half because it gives a quick and dirty estimate to her genetic contribution to the next year. It also has the convenient aspect that if you're a population ecologist and you wanted to look at the population growth rate, you attempt to do that by looking at the average survival of females in the population and half of their reproduction. In this case, because population ecologists tend to be interested in the female segment of the population, so you discount the number of pups by half under the assumption of equal sex ratios. So I was able to average across all the females, again remembering that we have fitness measurements on every individual in the population to get a population, or at least an estimate of land of the population growth rate. Not surprisingly, mean fitness did decline with emerging states. So the later the animals emerged from hibernation, the lower their relative annual fitness. I've just mean-centered emergent state here, and relative annual fitness is because I've divided by the population. And what has happened is the population itself has gone from one of growth in the first decade with most of the values above 1, to one of essentially maintenance, where it's hovering around a population growth rate of 1, which means replacement, essentially. And in fact, 4 of the last 9 years have actually had population growth rates below 1. And arguably, this phenological delay is somewhat more concerning for the ground squirrels because of its direction. When I talked about the fitness declines in response to phenological advances, typically those are due to the phenologies of the population not keeping pace with the phenologies of their food sources. Presumably, if those phenologies sped up a little bit, for example through microevolution, they would resynchronize and all would be hunky-dory. For ground squirrels, they're actually being pushed in the very opposite direction of what fitness would promote, or what would promote higher fitness. So if they were to go faster, it would actually accelerate the declines. And so undoubtedly, responses or phenological delays are less common in the literature than phenological advances. I predict, though, that we are going to start to see an increasing number of reports of populations that are affected more by, or at least equally by, precipitation as they are affected by temperature. And this is because global circulation models do predict for changes in precipitation in the same way that they do predict for changes in temperature. And specifically, over much of North America, and I fully admit that there's a lot of uncertainty in these models, but this shows one model up to the end of the century. Blue means it's getting wetter, brown means it's getting drier. And the consensus projections are for increased winter precipitation and increased heavy precipitation events over much of North America. So it's a leap. I definitely admit that it's a leap to use this as a causal explanation for what we've seen, but it does suggest that, or our results suggest that we have shown a link between the environmental variables, snow melt, and the phenology and the fitness of the individuals. So should such models come true, we can begin to predict the responses in wildlife populations that they're exposed to. So to conclude this section on hibernation phenology, by studying traditionally a physiological trait in a natural environment, we were able to show that it has ecological significance. When an animal emerges from hibernation, influences their individual fitness. When they emerge, is influenced by the environmental variables that they're exposed to. We sum it across the whole population. It influences the population dynamics. We're also able to show that it has some evolutionary significance in that it has an underlying genetic influence. It has some evolutionary potential. More generally, I just want to raise your attention or point your attention to something that's been up in the news recently that many of us have probably heard about. This is the spread of white nose syndrome in hibernating bats. This is an epidemic that was first seen in New York in 2006, and since then has swept across much of the northeast United States and eastern Canada, and is just about to knock on Texas's door. It decimates populations. Essentially, the animals become infected with cold-tolerant fungus, and it leads to massive die-offs in all the hibernaculum. So massive, in fact, that little brown bats are essentially the deer mouse of the bat world. Huge population densities, broad geographical areas, and there are calls to have them listed because they could go extinct in the next few decades because of white nose syndrome. And we know almost nothing about it. In fact, it was only just recently shown that this fungus actually causes the disease and isn't just a side effect of stressed bats, for example. One thing that we do know is that white nose influences hibernation patterns. This is the hibernation pattern of a healthy little brown bat. You can see it sort of having its periodic arousals to euthermy, just like the ground squirrels do. In a white-nosed bat, white-nose infected bat, the arousals are more common and more rapid. These arousals should be finely tuned so as to not deplete the fat resources of these hibernating animals. These guys are waking up regularly, essentially running out of fat and dying. So with Craig Willis at the University of Winnipeg, as well as some of his collaborators, we've started to look at the quantitative genetics of some of these traits and whether or not there's any individual variation in natural hibernation phenology as well as some other traits like body condition, habitat selection within the roost, etc., that could confer some resistance to white nose, and whether or not any of those traits are heritable, which could have some pretty major management implications. For example, if there is a trait that confers resistance to white nose, presumably we don't want to be interfering with natural selection. We want the fit phenotypes to make it through, and the poor genotypes to be washed out by the disease. Whereas, if these traits aren't heritable, or there is no link between white nose, more aggressive intervention may be necessary. So I see this as taking on a bigger relevance or a bigger direction in my lab as I move forward at Texas Tech, especially given some of the strengths, or the big strengths in that conservation ecology in the department. I'd love to talk to anybody else about some of our ideas, because we do plan on taking it to the field in the next step of the research. The next direction that I really see about my research taking is in trying to distinguish microevolutionary change from phenotypic plastic changes in response, or underlying phenology in response to climate change. As I mentioned a few times now, phenotypic changes are common in response to environmental change, but we really don't know how much of that is due to microevolution and how much of that is due to plasticity. And I was admittedly guilty of just doing that. I showed you, I sort of tweaked your interest, or piqued your interest a little bit by showing you that there's some evolutionary potential in the trait, in hibernation, emergence. Then I showed you there's been a phenotypic response. But what I didn't do was then start to disentangle those two things. How much of that might be due to microevolution? How much might be due to plasticity? And the reason for that is it's a lot more challenging than we might expect. And so Stan Booten and I were commissioned by the editors of a special issue of Evolutionary Applications that's coming up that is seeking to do just that thing, across basically all taxa. What is the evidence for these different things, microevolution versus phenotypic plasticity, in recorded phenotypic responses? And we've identified 3 approaches that can be taken: experimentation, inference from quantitative genetic models, and genomics. And these 3 approaches are essentially where my current research is lying and where I see my future research advancing to over the next 5 to 10 years. And I'll just touch really briefly on experimentation, and if I have time, I'll talk about quantitative genetics inference, and if I don't, I'm happy to talk about that later at the social something similar. So the experimental approach. One reason I'm working on bugging ground squirrels is not only do they have this really nice temporal record, or we have this really nice length of study, but they also have a really interesting distribution. And that's that they live in the Rocky Mountains of North America, all the way from the valley bottoms, all the way up right to the alpine. And presumably due to differences in the plant growing season in these 2 locations, or along this gradient, the hibernation phenology of the animals is also significantly different, up to a month later at higher elevations than at lower elevations. So what I've done is to take advantage of this natural variation in an experimental approach. Then I've moved squirrels from our low elevation site to our high elevation site, and the reverse. All of these photos, I should mention, were taken on the same day to give you a bit of an idea as to the climatic variation between my 2 study sites. And so by comparing the phenology of the animals that are moved to controls that went through this whole sort of soft release program or removed within the population, and also unmanipulated animals, I can start to gain some inference into whether or not there's genetic adaptation to the population, in which case I predict these animals will essentially hold on to their phenology even up there, versus whether it's more environmental variation, whether they'll adapt to that new population. These animals have now all been released and are coexisting with the residents. So we can record them under essentially natural conditions as long as they're strong. And I'll talk really briefly about just this other approach, a quantitative genetics approach to inferring evolutionary change. I'm going to jump systems a little bit here into some work that I'm doing with red squirrels, where they've also shown a phenotypic response to climate change. These guys are non-hibernating, they rely on cones to survive throughout the winter. And a previous account out of the lab that I'm working with now showed that they're showing the sort of typical response to temperature, and that parturition date is advancing in response, or over a period of about a decade. And they're able to go a little bit further, and what they were wanting to do is parameterize what we call the breeder's equation. It's called the breeder's equation because it was developed by animal breeders to document the effectiveness of breeding programs. Essentially, if you're wanting to select on milk yield in dairy cattle, you want to know how heritable milk yield is, you know what selection regime you're invoking, and you can predict the response to selection with those two traits, or pardon me, with those two pieces of information. As an evolutionary ecologist, we can do the same sorts of things by measuring the strength of selection and also measuring the heritability, as I've done for hibernation. They did that with the red squirrel parturition date when they're giving birth every year, and showing that the model predicted an evolutionary response. Whether or not it actually constituted an evolutionary response is a somewhat more challenging question. Specifically, that is because a compelling demonstration of an evolutionary response in a wild population needs to provide evidence of a direct relationship between the focal trait and fitness. And this is because, again, if we think back to environmental variation, you could have a squirrel that's sitting on a high-quality territory, they breed earlier every year, they have higher fitness every year. You might be tempted to conclude that there's some evolutionary potential or an evolutionary response due to that environmental heterogeneity and those environmental links. So the argument is that for there to be evolution in one trait, that trait needs to be genetically correlated with fitness. So any variation in that trait drags fitness along with it at a genetic level. In essence, there needs to be a genetic correlation or a genetic covariance between your trait and fitness. They also need to show that there has been a genetic trend in the population, and that that trait would be more than you just expect due to genetic traits. So I did this with our red squirrel data. It's a similar data set to the ground squirrels. It's about 25 years, and actually more animals, about 7,000 animals in this case. First thing I did was to look at heritability of parturition date, just replicating what Denis Rial had previously done. It is heritable. Second thing was to look at the heritability of fitness, same sort of thing, an animal model, quantitative genetics approach. Fitness is heritable in this case. But really what answered my question is that there is a negative genetic covariance, or standardized genetic correlation, between the two traits, suggesting that the animals that have the genes for earlier parturition, earlier birth dates, also have the genes for higher fitness. There is some evolutionary potential in this trait. The next step was to actually look at genetic change across time. And the way to do this was by using that animal model again, which provides you essentially two things. It gives you your genetic variance across the population, and it also gives you an estimate of the genetic merit of each individual, which we call a breeding value. It's a little bit of a foggy thing to get your head around, but essentially the way to think about a breeding value is if all other sources of variation are dropped down to zero. So there's no environmental variation, there's no maternal effects. An animal, this is hypothetical obviously, an animal that has a breeding value of -2 will breed 2 days earlier than me. That's the way to sort of encompass this. So essentially what I've done is, it wasn't as clunky as this, but essentially what I've done is do an animal model kind of each year and look at the breeding values across those years. This is what you'd expect just due to genetic drift. So this was essentially